Thank you. Okay, good. This young chap, Pete, has done all the work that I'm reporting. He's a PhD student, second year, a little after second year. So it's the results of about 2 years of quite hard work, most of which, of course, I won't have time to tell you. He was visiting here recently, so anybody in the world of methylation has probably heard a good deal of what I'm saying, but hopefully not all of it. I do have to acknowledge some of my philosophical mentors. I don't know if you know Yogi Berra, but he is famous for many, many quotes, of which one is that you can see a lot by just looking, and that's pretty much my theme for today. But we do have a model later, and we tried to make it as simple as possible, but not simpler. But as you will see, it was too simple. So we are now making it less simple, but that's another talk. Okay, so I really shouldn't be trying to tell you what methylation is, so I'll get over it pretty fast. You know, it's an epigenetic modification of DNA, and you know, it's important in gene regulation, development, cancer, lots of other things. And it's also important that it's preserved in mitotic division. And there's a picture of an embryonic embryo. Okay, this is very brief. There's unfortunately very little biology in this talk because there's very little time. But fortunately, I don't have to tell you about the biology. So you all know about 5-methylcytosine, the addition of the methyl group, and there's a picture of it, this guy, this CH3 thing losing its methyl there. And the important thing for what I'm going to talk about today is most methylation, at least in mammals I should add, is in a CpG context, and there's a picture probably not visible to most of you of a CpG context. And of course some regions have more CpGs than others. As some of you will know, not far from this place, about 25 years ago, 26 years ago, this was observed. Unfortunately, I have to say that I'm not using the local team's definition of CpG island. I'm using a group with whom we interact at the Hopkins. You know, and one could talk about why this might be more convenient. Why? But anyway, CpG islands means lots of CpGs together, and I will show you a picture about that shortly. So this is the one slide that has biology. Probably I shouldn't put any. But most CpGs are methylated, and most, of course, means in mammals, not necessarily in flies or bees or anything else like that. Highly methylated sequences include the following lists: repetitive elements, transposons, intergenic DNA, and exons. CpG islands are the exception to being mostly methylated, as they are mostly not methylated. And these, the reason this is that a lot of these CpG islands sit in the promoter regions of genes, and the majority of these are unmethylated. Sorry, that's not visible. I guess that's not my fault if it's hard to see. Maybe I should have put everything in the top half of the slide. And finally, just to make life interesting, a lot of these CpG islands get progressively methylated as time or other things go on. So therein lies the biological interest. But that's the end of my biology, folks. So if you're here for biology, feel free to leave quietly. How do we measure it? Well, there are lots and lots of methods, and people spend large parts of their life developing, refining, publishing these methods. I'm going to use some data from Ryan Lister, who collected this data while he was at the Salk Institute. It looks kind of old, but it is still a very large, publicly available set of data, which is what we wanted, as you'll see. Now, he calls, or at least these days, the way in which he's produced his data is called methyl-seq, but basically, which I do not have slides about, you know that you can treat the DNA with bisulfite, and then it does various things which I'm not going to spend time on, and then there's a lot of stuff that I'm not going to spend time on. A lot of careful bioinformatics is needed, mapping these reads to the genome, but at the end of the day, you get some sort of data about genome-wide methylation of the cells you started with. Now, start with a cartoon because then I can understand it. If you think of a chromosome, and then you know, just focus on the CpGs and put them one by one. Of course, they're not equally spaced, and you put a little black line, a black dot if they're methylated, and a white dot if they're not. Then you might loosely think this is just made-up data. And then, of course, there's lots of these things. And equally, we do not get the entire thing. We get little segments of them when we do this bisulfite treatment followed by sequencing. But we can look at it, and I'm going to show you some looking at it. As you can see here, that's a pretty high resolution picture, single base resolution down the bottom there of a gene. If we look a bit bigger there, you can see the 5' UTR of the gene, and you can see the little hatches there, which are the CPGs. That's almost 4 kb, and that guy fits somebody's definition of a CPG island. Seeing this is of a browser, it will not be the one I'm using. It'll be the Santa Cruz definition, which is the local definition. If you now look at your data, these are the reads, forward reads at the top, reverse the other direction, and you label the CPGs red if they're methylated and blue if they're not. That's a sort of snapshot of the data we have to work with. And then if you look at this, 2 kb there, here's our CPG island again and our gene, and you can see that you can see it's mostly methylated, contrary to what I told you earlier, where this is probably encompassing the promoter region of that gene. Pull right back and you see this is now almost 30 kbs. There's a little spot over there where there's a CPG island. In another cell type, it's actually unmethylated, but in the first one, it's methylated. So this is the sort of data, and there's a lot of work there, really months and months of hard work by Pete to organise the vast amounts of data that Ryan Lister put on the web, which I'm going to pass over. Measuring aggregate methylation. This is not a commonly used term, but you'll see what I mean by it in a minute. So here is my sort of collection of little cartoon strings. I'm going to call them from now on haplotypes. So they're really, one of the 2 chromosomes, of which, forgetting the X and the Y for the moment, we should have 2, and we can look at the methylation status of the CPGs. Now, in some sense, if you wanted to know the average methylation at a particular CPG, you would just sort of add up all those guys along all the chromosomes that were in your cell sample. But of course, we can't do that. What we can do is do a similar sort of thing with all the reads we've got. So if these reads are proportional in abundance to the actual haplotypes in your cell sample, then these averages here that give you numbers like 6 out of 10 or 1 out of 13 will be some sort of estimate of what I'll call the aggregate methylation at a particular position. And we're going to call them beta, the Greek letter beta. Okay, so that's a measurement. That's the number of methylated sites divided by the total number of sites, methylated plus unmethylated. Now, what do the betas look like? There's a histogram there, which shows quite a spike. I'll explain the colours in a minute. It's got a spike near 0, a spike near 1, and a bit of stuff in between. So a lot of stuff's methylated, a lot of stuff's unmethylated, and there's some in the middle. And that's a histogram there where the bin size is 1%. So we're going up from 0 to 100% in 1% intervals. Well, there's 2 sorts of colours there. The blue is the CPG islands, which you can see are mainly down the left-hand end, mainly unmethylated, and the red is everything else, and they're mainly methylated, but a bit in the middle. So that's a sort of typical distribution of methylation, this aggregate methylation number that I'm talking about. Now I'm ready to tell you what you're sitting here to listen to. Co-methylation. What is it? Well, loosely, it's the persistence of methylated or unmethylated, the persistence of the state along a haplotype. Think chromosome, but haplotype just sort of drives it home. We're not interested in the pairing at this point. Slightly more precisely, the association between the methylation and the unmethylation at 2 particular sites separated by some distance, which we're calling IPD, interpair distance. Take a pair of CPGs at a particular distance. How associated are the methylations, the methylation states or the methylation measure? It's meant to make you think of correlation, of course. And I want to tell you very briefly why we study it. One is that we were studying it for a statistical reason, and we haven't got back to that yet. And Pete will probably finish his PhD never getting back to the reason we started studying it, but we hope that's not going to prevent him getting a PhD. Then we got interested in it in its own right, which is why I'm standing here. And then we discovered there are a small number of biologists on the planet who are also interested in it in its own right. Co-methylation, this is, not just methylation. There's millions of people interested in methylation. Exaggeration. Tens. Hundreds. But there's not that many people interested in co-methylation. But then we got a puzzle, which we have now been puzzling over for well over a year. Still haven't got there. So this is the motivation. This is a slide produced, or at least a figure from a paper, 2011, former Berkeley student who went to Hopkins to work on methylation. And he's figured out a way of identifying differences, differential methylation. In the top panel, you can see we have some blue lines. That's 3 normal tissues, and the slightly below it is 3 tumour tissues. Matched, actually, from the same samples, though that's not terribly relevant. And you can see that in general, the methylation level, this is that beta, this aggregate methylation, that's a 2.5 megabase region. So that's a very big region of the genome, or very big by some people's standards. And a similar thing down the bottom there. So they have a procedure for identifying differential methylation between tissues. You might also want to identify regions in the same genome that are differentially methylated. And when we asked them, well, how are you dealing with the issue of correlation or persistence of methylation along the chromosome? The answer was, oh, we do blah-de-blah-de-blah. But the short answer was, we don't know. There wasn't a lot, there's nothing built in about whether methylation might last 10 kb, 100 kb, whether it might disappear, at least the signal might potentially vanish after 50 base pairs. So that's what we got interested in looking at. Okay, so I'm going to talk now about a cross haplotype, this aggregated co-methylation. If we have some interpair distance here, how similar are the aggregate methylation values at typical CpG pairs separated by that distance? Well, the first thing to do is you might say, do a scatter plot. Now, if you think hard for about a minute, you'll realise that you can't have 2 CpGs 1 base pair apart. They have to be at least 2. So this is as close as it gets. And that's a kind of a fuzzed version of a scatter plot with literally tens or hundreds of millions of dots. Not hundreds of millions, well, okay. You know how many CpGs there are in the genome. They're pretty much all there. And of course, there's pretty high correlation, mainly because they're either both 0 or both 1. Right? They're either go out a bit further, correlation goes down, 250 base pairs. Go further again, 20,000 base pairs, still a correlation almost 0.1. Now, I'm sure anybody who did a course in statistics knows that you're supposed to reflect deeply on the issue of whether correlation implies causation, and whether you think that whatever it is that is making methylation here might be causing it to happen to some level 20 kb away, or whether it's something else. So that's part of our puzzle. So here are some summaries of the beta values. This is the first one, out to 500 base pairs in one of Ryan Lister's cell lines. Pearson correlation on the vertical axis, interpair distance on the horizontal axis, and we are going from 0 to 500 base pairs. And there's one, which is, of course, complete, well, it can't be achieved, but pretty close. And the green is the CpG islands. So just focus on the green for the moment. CpG island correlation, it stops at about 300 because, well, I'll explain. We have eliminated, we're only looking at things that are that far apart with nothing in the middle. Right? So when you say we take 2 points, 2 CpGs separated by a certain distance, that could come in 2 ways. There could be lots of CpGs in the middle, or there could be none. This is the picture where there's none. So you're not going to find too many CpG islands where there's supposed to be lots of CpGs which have 2 CpGs miles apart. That's sort of contradictory to the idea of there being lots. So that stops. Whereas if you go outside CpGs, you can go on for a long way. And there's an interesting little sort of periodicity there. Anyway, that's the picture. The correlation holds up. This, if we stretch it out now to 2,500 base pairs, the CpG story's died because we've run out of, this is removing the stuff in the middle, and the other one holds up. We're still sitting there at about 0.2 at 2,500 base pairs. And if we go out a long way, which I'm apologising for it getting rather noisy, you can see this is now 25 kb, 25,000 base pairs, and just focus on the CpG islands now. Here we have left out the requirement that there's nothing in the middle. But you can see that this blue line, for example, non-CpG islands, is just below 0.2 all the way out to 25,000 base pairs, which is part of our puzzle. So I'm going to summarise those observations. We see a periodicity. I didn't show you the periodicity of 10 because I would have had to put another slide in with a higher resolution, but it's there. And you see about 170 in the Pearson correlation. We're not the first people to see this, but we do see it pretty clearly. It varies according to genomic context and cell type, somewhat, but there are common features. But the non-zero correlations extend right out. See, I'm almost finished, according to him. Slide 34. Another 20 slides. Just joking. Slightly. Okay, so now we've got to get to the other thing. I want to get to my puzzle, the within haplotype correlation. So now I'm going to look that way, within a chromosome. And we do it, see, if I take, this is now, if we look at these guys, we can find some on the same strand. So obviously that's within a chromosome. So what we're going to do is pick all the guys that have 2 CPGs and nothing in the middle on the same fragment, and count them up. There's 1 which is methylated in both, there's 2 which are methylated in the first and not the second, and none the other way around, and 4 that are unmethylated in both. And we compute a number called the log odds ratio, which is the 2x2 table equivalent of a correlation coefficient. We do not compute Pearson correlations in 2x2 tables. We compute this guy for reasons I could tell you later. And this is, of course, separated by, whoops, what's happened? Separated by 44 base pairs, and we have to do this at all separations where it's feasible. Now, here's a picture of what you get. The red line, they've been separated by 2. The green line separated by 20. The blue line by 60, and the purple line by 120. And these are these log odds ratios that I'm talking about, and you see the red line there centered around rather a high value, which means a very strong association, whereas as you get greater separation, it shifts towards the left. These purpley ones, they're not quite centered around 0. 0 corresponds to independence, no association at all. But it's heading that way. Now, these are individual pairs where we have multiple reads, and we can compute, if you like, pair-specific log odds ratios. If we aggregate across all the pairs of a particular interpair distance, we can get this picture. This is out to 80, and that's a different cell line in Ryan's list. And you see, we see a nice 10 base pair little bump, but that's not terribly important here. But anyway, that's the sort of thing. And you see it goes down from 4, which is pretty high, and it's still up at 1, which is not 0. 0 is independence, but it's heading there. If we go out a bit further, which we can do because Ryan's data is paired-ended, so you can actually look at CPGs at each of the 2 pairs, then they have a reasonable separation. We're not within a read, we're between 2 reads. And we go out now to 300, and you see it's heading towards 0. We haven't got anything much beyond 300 because it's hard to get CPGs that are in the same strand, at least with this Illumina. This is 75 base pair paired end reads. We could obviously go to a different platform, but then we'd have to get somebody to do this. Brief summary. Interesting patterns that are visible in the log odds ratios. The same 10 base pair cycle. An interesting feature about 200 base pairs. Interesting that it sort of comes up a little bit around about there. The pattern of dependence actually changes markedly with genomic context and cell type. Don't have time to show you all this. But in most cases, the dependence is high and has almost vanished after a few hundred base pairs. So, the inconsistency here is that if you look within a strand, the dependence almost vanishes after a few hundred. If you look at the aggregate measure, it's sitting there at 0.1, 20,000 base pairs apart. So we thought we'd better try to understand why that is, what is the cause of that difference. Okay? Well, this is the first failed effort to understand this inconsistency. Our model was too simple. Einstein would have said, Terry, my boy, I said, simple but not too simple. So, well, we didn't know until we tried. So this is a very quick picture of, I haven't got time to give the details because he's going to tell me it's 5 minutes pretty soon. But anyway, we had a particular simulation model. And just as a side comment, the reason we wanted to do this for having methods for detecting differential methylation is that statisticians like to simulate the data they're analysing so they can test their methods on simulated data to see how it works when you know the answer. The trouble with so-called real data is you never know the truth. So how can you tell whether what you call differentially methylated really is differentially methylated? The answer is simulate. But of course the simulated data has got to look like your real data, or otherwise it's not much point in testing in that context. So these are ways of simulating which I won't spend time doing, and this is a simulation procedure. And as it's now clear to us, part of the problem is there are multiple haplotypes in a given cell sample. I mean, there's often multiple cell types as well, but even if it's the same cell type, because after all, these cell lines are cell lines. They're not supposed to be mixtures of cell types, but they do not just have two haplotypes, the one from mum and the one from dad. They often have lots of haplotypes, presumably because as time goes on and you have the replication of the cells and the methylation's laid down or it's meant to be copied, errors are introduced, and so on and so forth. So we simulated a diploid methylome in two ways, one by just taking independent haplotypes and the other by giving the haplotype a father or a parent and then copying them. I won't go into details here. But here's what we got. See, this is the distribution which you saw earlier of the genome-wide distribution of beta, the aggregate methylation, and that's what we found in our simulation. Of course, not surprising, we got the proportions of unmethylated and methylated pretty right. We could hardly get anything else. So we obviously didn't do it right to get this sort of stuff. So that wasn't great. Our beta correlations didn't look like the real data either. They came down to zero much, much more quickly, which makes sense. And by the way, this simulation that I've described builds in the within haplotype simulation as we saw it, a particular version of it. If you look out to 25 kb, we have nothing. The real data has lots. So this is sort of a comprehensive failure. You can't actually see, I'm afraid we didn't have time to get, I got the last revised versions of these figures about 10:30 last night. Poor old Pete was not feeling too well, so he went to bed before he could actually make that picture. There is some data here which, the main thing is they're different. Okay, so this simulation did not work. So we're moving on, following Einstein's dictum, to a less simple model, which we hope is not too simple. So we're going to have multiple haplotypes. We did not have that before, and this was a challenge. How do you estimate the number of haplotypes in a sample of cells? If you want to think about that for a microsecond, it's not obvious. Their numbers and proportions vary. We had to reflect a little bit more than CPG and non-CPG, and people bang on about partially methylated domains being the big thing. So we got partially methylated domains according to one expert's definition, and we got the rest, and the rest we've divided into unmethylated, lowly methylated, and mostly methylated. And we've got region-specific features, that is, what sort of associations along the haplotype we build in, because they do differ, whether they're coming from a partially methylated domain or a mostly methylated domain or an unmethylated domain, and so on. Now, and then there's also an issue of how do we capture the dependence along the chromosome? Because you can have quite large CPG islands where everything is unmethylated, and that is a super strong dependence, which is pretty hard to capture statistically in what we call a one-step model. So that's our lots of simple model. Will it give realistic plots where the simple one failed? And the answer is, we do not know yet. And that's the end. So there's Peter, who did all the work. And there's Peter Hall, who is his supervisor when we get onto the problem that we started on, but we haven't got there yet. And there is the person who dragged me into this fascinating field. So she introduced me to the mistress of the genome, and I'm having a good time. And here's some of my colleagues from Baltimore, all of these two of whom are ex-Berkeley, so I taught them in Berkeley, and now they're doing methylation in Hopkins. And we've had various help from people at other centers of excellence of methylation, namely the University of Southern California and the Babraham Institute in Cambridge. I thank you for listening.